Welcome back to Bite Size MCAT. Let's take some time to practice enzyme inhibition problems in a variety of different contexts that you might see on the MCAT. Take a look at this problem. The enzyme acetylcholinesterase follows Michaelis-Menten kinetics. In the presence of an inhibitor, the enzyme's Vmax stays the same, but the substrate concentration needed to reach half Vmax doubles. Where did this inhibitor bind? Go ahead and pause and take some time to think through this question. Okay, let's walk through this step by step. What information does the question actually give us? So first, we know that Vmax stays the same and that the concentration needed to reach half Vmax doubles. Remember that by definition, the concentration needed to reach half Vmax is known as Km, and this increases as it doubles. So what type of inhibition is this? Well, the two things, Vmax being unchanged and Km increasing, means that this is competitive inhibition. Okay, great. So now let's think about where a competitive inhibitor binds to. A competitive inhibitor binds at the active site, which is only found where? Yeah, on the free enzyme that's not being bound up. So the answer here is A, the free enzyme only. What about the other answer choices? Well, the free substrate only is actually referring to what the enzyme itself can bind to, so that can't be right. B can't be right. The enzyme substrate complex only is what an uncompetitive inhibitor binds to, so C is incorrect. And both the free enzyme and enzyme substrate complex is what a mixed or non-competitive inhibitor would bind to, so we could rule that one out as well. Now, try a slightly different wording for a problem. These are to kind of get you used to how the MCAT might ask about this content in passages. So the enzyme glucokinase catalyzes the phosphorylation of glucose. A researcher adds an inhibitor that binds to a site distinct from the enzyme's active site. There is equal binding affinity for both the free enzyme and enzyme substrate complex. Which of the following best describes the effect of this inhibitor on enzyme kinetics? Take some time to think about this one. Okay, now note what we are given in the question. First, the inhibitor binds to a site distinct from the enzyme's active site, meaning it is some sort of allosteric site, not the competitive site. So therefore, this can't be competitive inhibition. We are also told that this binds equally to both the free enzyme and enzyme substrate complex. So what inhibitor does these kind of things? Yeah, a non-competitive inhibitor. So now recall what happens to the kinetics with the addition of a non-competitive inhibitor. Well, Vmax decreases and Km stays unchanged. So which one of our answers shows this? Well, yeah, that's B. What about the other answer choices? Well, let's first look at A. Km increasing and Vmax decreasing is actually an example of mixed inhibition, where an inhibitor has different affinities for free enzyme and enzyme substrate complex. C says Km decreases and Vmax decreases. And what is this an example of? Yeah, this is uncompetitive inhibition, so not non-competitive inhibition. Now, what about Km increasing and Vmax being unchanged? Yeah, that's competitive inhibition. So all of this leads us to believe that B, Km being unchanged and Vmax decreasing is the right answer for non-competitive inhibition. How about one last practice problem? So a researcher gathers data about an enzyme with and without inhibitor and generates the data above. Which of the following graphs depicts this kind of inhibition? Pause and take a moment to answer this. So this is a great way to apply the knowledge we've learned and a real possibility for a type of data analysis question on your MCAT. Observe what data we are given in this table. Km goes from 16 millimoles to 8 millimoles with the inhibitor, so that means it decreases whenever we add the inhibitor. Vmax similarly goes from 14 micromoles per minute to 7 micromoles per minute. This means it also decreases. So we know that Vmax and Km both decrease. So what kind of inhibition does this? Yeah, it's an example of an uncompetitive inhibition. So what type of graph do we have with uncompetitive inhibitors? If you remember back, uncompetitive inhibitors have this unique structure in which we have parallel lines. So our correct answer here is going to be D, the parallel line graph. So what about the other answers? A shows Km decreasing and Vmax increasing. So what type of inhibition is this? Yeah, it's mixed inhibition. What about answer choice B? B shows Km increasing and Vmax staying unchanged. So what type of inhibition is this? Yeah, it's competitive inhibition. C shows Km staying the same while Vmax decreases. How about this one? Yeah, it's non-competitive inhibition. Great job. So I hope that all of these questions and explanations helped and made sense. Thanks for watching.